The overall goal of this procedure is to perform 3D X-ray microtomography of mineralized and non-mineralized tissues. This is accomplished by first extracting the samples to be examined. Then immobilize and position the samples in the micro-CT instrument. The third step of the procedure is to set acquisition parameters for each sample and collect the images. Ultimately, a 3D image of the sample at high resolution through X-ray CT is obtained.
The main advantage of this technique over existing methodologies like MRI, ultrasound, or electron microscopy is that it provides information about thick volumes at 1 micron resolution. Though this method can provide insight in many biological tissues, it can be used also in electronics, in the materials sciences, and in archaeology.
Preparation is different for mineralized and non-mineralized tissue. Mineralized tissues must be sealed in containers with a tight fit, so sample position does not change while high-resolution measurements are made. The container shape will vary according to the tissue's morphology. For example, a mouse femur extracted from an 18.5-day-old embryo can fit into a pipette tip. Begin by sealing the narrow end of a polystyrene pipette tip with glue, like epoxy resin. Then, fill the tip with a working buffer, in this case PBS. Now, fit the leg with exposed femur tightly into the tip